So hello everyone, my name is Neha Shada. Today I will teach you molecular basis of inheritance. Okay, by seeing this slide you already got to know. Today we will learn molecular basis of inheritance. Okay, so in this chapter we will learn structure of DNA. Okay, structure of DNA and what makes DNA our genetic material. Okay, so let's get started class. Molecular basis of inheritance. What do you mean by the name? First of all, let's understand molecular basis of inheritance. Inheritance means it is the process by which our genetic material transfer from parent to offspring. Okay, parent to offspring. Okay, so our eyes look similar to our parents. My eyes look similar to my father's eyes. Okay, your eyes look similar to your mother's eyes. Our hair color is similar to our parents. So what is the reason behind this? Okay, so there is something we call DNA, which is genetic material of human beings, or it is the genetic material of many living organisms. Usually DNA is genetic material of living organisms except some viruses. Okay, in viruses RNA is genetic material. Okay, so we got this genetic material from our parents. Okay, I got DNA from my parents. Okay, 50% from my mother and 50% from my father. Okay. So in this chapter, inheritance, we got to know what is inheritance. It is the process by which our genetic material transfer from parent to offspring. Inheritance. Okay. What is genetic material? Genetic material in living organisms can be DNA or RNA, which stores their information. Okay, which stores the information. Stores information. Okay. So, in this chapter, we will learn the structure of DNA at molecular level. Okay, molecular basis of inheritance. The name suggests that we will learn structure of DNA, RNA, how DNA is our genetic material, how it gives information. Okay, why DNA is our genetic material, how it transfers. Okay, why only DNA, not protein or RNA. Okay, we will learn structure of DNA at molecular level. Okay, so structure of DNA is discovered by 4 scientists. Okay, Watson and Crick. Watson and Crick are 2 scientists who give structure of DNA in 1952. Okay, they simply give structure of DNA using the data given by Rosalind Franklin and Wilkins. Okay, Rosalind and Franklin are main scientists who use X-ray crystallography to give structure of DNA. Okay, so Rosalind Franklin and Wilkins give structure of DNA. They uses the DNA structure looks like this. Okay, in X-ray crystallography. Watson and Crick use this data, okay, given by Rosalind Franklin to give structure of DNA. Okay, so in 1952, Watson and Crick collect data and give structure of DNA. Okay, so let's study next. So who gives structure of DNA? Watson and Crick gives structure of DNA in 1952 using data given by Rosalind Franklin and Wilkins. Okay. And in 1962, Watson and Crick and Wilkins got Nobel Prize. Okay, now you are wondering why Rosalind Franklin didn't got Nobel Prize, because she died before getting Nobel Prize. Okay, she died before 1962. That is why only Watson and Crick and Wilkins got Nobel Prize for this amazing discovery, which is structure of DNA. Okay, so what is structure of DNA? Okay, what is structure of DNA? So DNA has three main components. DNA is made up of three components that are nitrogenous base, phosphate, and pentose sugar. Okay, both DNA and RNA. Okay, there are some differences between DNA and RNA which we studied in upcoming slides. Okay, so these nucleic acid, we also know, we called it as nucleic acid. Okay, they are made up of nitrogenous base, phosphate, and pentose sugar. Okay, DNA. So what is nitrogenous base? So we have two types of nitrogenous base. That are purines and pyrimidine. Okay. Purines are 9-membered aromatic structure. Okay, it is look like this. And pyrimidine look like this. It is 6-membered, it is 9-membered ring. Okay. Purines are of two types, adenine and guanine. Okay, and pyrimidine are of three types, uracil, thymine, and cytosine. Uracil is present only in RNA. Okay, uracil is present only in RNA and thymine is not present in RNA. Okay, so here we will discuss what are the nitrogenous bases which are present in DNA and RNA. In DNA, adenine is present, guanine is present. In RNA, both purines, adenine and guanine are present. And in case of pyrimidine, in DNA, thymine and cytosine are present. And in case of RNA, uracil and cytosine are present. Okay, so in RNA, thymine is replaced by uracil. Okay, so this is difference between DNA and RNA. This is first difference between the nitrogenous base. Second component of our nucleic acid, DNA or RNA, are pentose sugar. What is pentose sugar? Pentose means 5-carbon sugar. So this is how the structure look. And from this structure, we will learn the second difference between DNA and RNA. Okay, second difference between DNA and RNA. So if I ask you what is the full form of DNA? DNA stands for deoxyribonucleic acid. Okay, deoxyribonucleic acid. And RNA stands for ribonucleic acid. Okay, ribonucleic acid. So in in RNA, ribose sugar is present. Okay. And in ribose sugar, if you see the structure very carefully, you will see a difference between ribose and deoxyribose sugar. In case of RNA, we have an extra hydroxyl group at position 2. Okay. At 2' carbon, there is an extra hydroxyl group. Okay. Other things are same, but we have an extra hydroxyl group at 2' position. And DNA stands for deoxyribonucleic acid. It also has ribose sugar, but the oxy group at 2', okay, this hydroxyl group at 2' is absent in DNA. Okay, this hydroxyl group at 2' is absent in DNA. That's why RNA is much unstable as compared to DNA. Okay, DNA has hydrogen and hydroxyl group is not present here. Okay, OH group is replaced by hydrogen. That's why its name is deoxyribonucleic acid. Okay, and RNA is ribonucleic acid. And this hydroxyl group makes RNA very, very unstable. Next is phosphate. Third is phosphate. So this phosphate will attach with ribose sugar. Okay, ribose sugar. And this phosphate and ribose sugar make sugar phosphate backbone and DNA. How it's made, we will see in upcoming slide. This sugar and phosphate forms sugar phosphate backbone. Okay, how it makes backbone, what is this backbone, we will learn in next slide or upcoming slide. Okay, so what are components of DNA? Let's revise. The components of DNA are phosphate, nitrogenous base, and deoxyribose sugar. Okay, and if I ask component of nucleic acid, so these are components of nucleic acid: phosphate, nitrogenous base, and pentose sugar. And we have two types of nitrogenous base: purine ring and pyrimidine ring. Purines are of two types: adenine and guanine. Pyrimidine are of three types. What are the? Thymine, cytosine, and uracil. Okay. So clear class? Pentose sugar, nitrogenous base, and phosphate. Okay. Now we have to make bond between them. Okay, they have to attach with each other. They have to form bond to make DNA. Okay. So nucleoside. First we will form nucleoside. This ribose sugar will form bond with nitrogenous base. Okay, will form bond with nitrogenous base and form nucleoside. Okay, nucleoside. This bond is known as N-glycosidic bond. N-glycosidic bond. Okay, the bond between pentose sugar and nitrogenous base is known as N-glycosidic bond. It will form a nucleoside. For example, adenosine, guanosine, okay, cytosine. If adenine is present here, if guanine is present here, okay. So nitrogenous base will form bond with carbon number 1. Nucleoside will form. Now, with this nucleoside, then a phosphate is attached. Okay, nucleoside is pentose sugar and nitrogenous base. Okay, sugar and nitrogenous base. When to this nucleoside phosphate group is attached. Phosphate group will attach here. Okay, phosphate group will attach here. When phosphate is attached, it will form nucleotide. It will form nucleotide. And nucleotides can be nucleotide monophosphate, nucleotide diphosphate, or nucleotide triphosphate. Okay. One example of triphosphate is ATP, adenosine triphosphate. Okay. When a phosphate is attached with nucleoside, okay, it will form adenosine triphosphate, which is a nucleotide. Okay. And the bond is phosphorester bond. Okay. So here is our nucleotide, a nitrogenous base. Okay. Bond is, what is this bond? And glycosidic bond. What is this bond? And glycosidic bond. And what is this bond? Phosphoester bond. Okay, phosphoester bond. This is phosphoester bond. Okay, and this is our nucleotide. Now this nucleotide will attach with each other. This is like adenosine. What is adenosine? Adenosine is our nucleoside. Many students confuse between nucleoside and nucleotide. Nucleoside is only sugar and nitrogenous base. When phosphate is attached with it, it will form nucleotide. Okay, nucleotide. Only one phosphate is attached through phosphoester bond. It is known as adenosine monophosphate. When two phosphate group will attach, adenosine diphosphate. When three phosphate attach, adenosine triphosphate. Okay. Now, these nucleotides, we also know that DNA or nucleic acid are known as polynucleotide. Polynucleotide. Okay. So these nucleotides, okay, first nucleotide and second nucleotide must have attached with each other. Okay. So must have to form bond. Okay. So they will form phosphodiester bond. Okay. And the bond will form between, for a second. Bond will form between 5' phosphate. You have to remember the step. 3' hydroxyl group. At 3 carbon, at third carbon, hydroxyl group will present. At 5 carbon, phosphate group is present. Okay. Like this now. At 3 carbon, hydroxyl group is present. At this carbon, CH2, a phosphate group is present. Okay, phosphate group is present. Okay. So, the bond will form between this 3' carbon, 3' hydroxyl group and 5' phosphate. Okay, this bond will form. It is very important. You have to learn, or you don't have to cram this. You have to understand that at 5' position we have phosphate group and at 3' position we have hydroxyl group. Okay. Now, this hydroxyl group of 3' and this phosphate group at 5' position of ribose sugar will form phosphodiester bond. Okay, this is condensation reaction. Water will remove. Okay, this water will remove and phosphodiester bond will form. Okay, 2 ester bonds. Here is only one ester bond. It is phosphoester bond, and 2 ester bond, phosphodiester bond will attach to nucleotide. 2 nucleotide is attached. Okay, the reaction will continue. Okay, the reaction will continue. At 3' hydroxyl group will attach with 5' phosphate. Okay, and it will form polynucleotide. Okay, it will form polynucleotide. So these polynucleotide will form a long DNA. Okay, this polynucleotide will form a long DNA like this. Okay, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay, so at 5' position we have phosphate, at 3' we have hydroxyl. Okay, so this sugar phosphate will form backbone of DNA. Backbone of DNA. Now, these nitrogenous bases which are attached to sugar will form hydrogen bond with another long chain, with another polynucleotide. This is our first polynucleotide chain. This is our second polynucleotide chain. Now, these will attach with each other with the help of nitrogenous base. Okay, so nitrogenous base will form hydrogen bond. Adenine is what? Adenine is purine. Okay, adenine will form bond with thymine, 2 hydrogen bond, and guanine will form 3 hydrogen bond with cytosine. Okay, so these are hydrogen bonds, and you also know that polarity. DNA have polarity. If one strand is from 5' to 3' direction, the other strand will 3' to 5' direction. Okay. Okay. So at 5' we have phosphate group, at 3' we have hydroxyl group. Okay. And this through this opposite polarity, DNA will form hydrogen bonds. Okay. So in this diagram, if I ask you how many bonds are present, first hydrogen bond between 2 nitrogenous bases, second N-glycosidic bond between sugar, ribose sugar and nitrogenous base. This is N-glycosidic bond. And this is phosphodiester bond. Phosphodiester bond. Okay. So this is how a long chain of DNA polynucleotide will form. Okay, 2 polynucleotide chains will form hydrogen bond, bases forms hydrogen bond, and this DNA will form helix. Okay, these 2 DNA chains. Okay, these 2 polynucleotide chains will form a helical structure. Okay, and the polarity is opposite. One strand is from 3' to 5' position, means here we will have 3' hydroxyl group and here we will have 5' phosphate group. Okay, so you can also see in diagram 5' phosphate group, 3' hydroxyl group, 5' phosphate group, 3' hydroxyl group. Okay, and adenine and thymine has 2 hydrogen bond, guanine and cytosine have 3 hydrogen bonds. Only guanine will form bond with cytosine. Okay, adenine will form bond with thymine and guanine will form bond with cytosine. Okay. Now, another important thing about DNA, they will form a helical structure. Helical structure with opposite polarity. One is from 5' to 3', another strand will of 3' to 5'. Now, these helical structures have diameter of 2 nanometer or 20 angstrom. Okay. The diameter is 20 angstrom and one helical turn have 10.4 base pairs. Base pairs are nitrogenous base. Okay. One helical turn has 10.4 base pairs. Okay. And the distance between 2 base pairs is 0.34 nanometer. And this one helical turn will 3.4 nanometers. Okay. Or 34 angstrom. Okay. So this is our DNA. This is our sugar phosphate backbone, which must now it must clear to you how this sugar phosphate backbone forms. Okay, and wait for a second. Now these nitrogenous base will form form at center, and this one helical turn is 3.4 nanometer, contains 10 base pair, 10.4 base pair. Okay. Now this is the structure of RNA. RNA is usually single-stranded RNA, but sometimes RNA can also be double-stranded. Okay. RNA can also be double-stranded. Okay. So this is structure of RNA. The difference between RNA and DNA is RNA have uracil and RNA have 2' hydroxyl group. Okay. This 2' hydroxyl group is very reactive. That is why evolution choose DNA over RNA. Evolution choose DNA over RNA. Okay. So this is structure of RNA. Okay. Now why DNA? This is structure of DNA. Okay. DNA have nitrogenous base, phosphate sugar, phosphate and sugar. Okay. But why these three components forms DNA are genetic material? Why DNA are genetic material? Okay. And why our DNA is not similar to chimpanzee DNA, or my DNA is not similar to your DNA? Okay. The structure will be same, but the difference between my DNA and your DNA is sequence. Okay. The sequence of nitrogenous base. Sequence of nitrogenous base. Okay. So if this is 5' to 3', this is 3' to 5' strand of DNA, this will have a sequence A, T, U, not U, A, T, C, G, A, T, C, G. Okay. And in another strand, they are complementary to each other. Other strand will have T, A, G, C, T, A, G, C. Okay. There will be a sequence in DNA. Okay. These sequence will decide what will be the sequence of RNA. Now RNA will have U, T, U. Okay, not U. RNA will have A. Okay. T. Instead of T, we will have U. Okay. C, G, A, U, C, G. Okay. There will be a strand of RNA. Okay. And from this RNA sequence, we will get protein. Okay. So who will decide what kind of protein will form? This sequence. Okay. This sequence will decide what kind of protein is formed. And protein is our building block. Okay. Protein are building block. Okay. So that is why DNA is our genetic material. DNA stores our information. Okay. The sequence of DNA stores our information, and this information will change into first RNA. Okay. RNA give rise to proteins, and proteins are our building block. Okay. Any change in the sequence of this DNA, will give rise to mutation. Okay. Any change. Okay. So this is central dogma. DNA will form another DNA, which is known as replication. Okay. From DNA, RNA is formed, which is known as transcription. The process is known as transcription. And from RNA, we will got protein, which is the process is known as translation. Okay. Any change in the sequence of DNA, will form a non-functional protein. or wrong protein. Okay. So that is why the sequence of DNA and transfer of this DNA from parents to offspring is important, and DNA is our genetic material. It stores that information. Okay, it stores that information in the form of these sequence. Okay, and this sequence is different in different organisms. Okay, and this sequence will decide the sequence of protein. That is why any change in this sequence will cause mutation. Okay, any change in the sequence of DNA will cause mutation, and we will got our wrong protein. Okay, so from DNA, from 2 strands of DNA, we got RNA through transcription, and from transcription, we got these are 3 codons. AUG codes for methionine. This is a protein, amino acid. Then B sequence TCG will code for proline. Okay, and similarly, these amino acids will form proteins, which are our building blocks. Okay, that's why the sequence of DNA is important, and DNA is our genetic material. Okay, so with this we will end this class and this session. Let me summarize this session for you. In today's session, we learned structure of DNA. Okay, with structure, molecular structure. Okay, molecular structure. But it's the structure of DNA composed of nitrogenous base, phosphate, and deoxy sugar. Deoxyribose sugar. Okay, and RNA has ribose sugar. Okay, and difference between RNA and DNA. Plus central dogma. Okay, so with this we will end this class. If you have any doubt, you can ask me. Okay, so bye class. Have a good day. Thank you.